Okay, so we're going to cover the last step in cell respiration for aerobic cell respiration, and that's oxidative phosphorylation. So you remember we had glycolysis happen in the cytoplasm, right? And what we got out of glycolysis was 2, 2, and 2, right? 2 ATP, we got 2 electron carriers, NADH plus H plus, and we got 2 pyruvates, right? And then those pyruvates, I'm just going to draw the mitochondria here now. I'm going to draw this one over exaggerated in the... Okay, so then that pyruvate will be transferred in the link reaction, right? And in the link reaction, we get an NADH plus H plus for each pyruvate, right? So since there's 2 pyruvates, I'm just going to put a little 2 there, right? And then that's going to go into the Krebs cycle. And in the Krebs cycle, we end up getting, and I'm going to do this for the glucose. So we get 6 NADH plus H plus, we get 2 FADH2s, and we'll get 2 ATPs. Okay? All right, glycolysis, link, and Krebs cycle. Oh, one thing that's important too is this is called the matrix. The Krebs cycle happens in the matrix of the mitochondria. So glycolysis is in the cytoplasm, right? And the Krebs cycle happens in the matrix of the mitochondria. And the last bit, oxidative phosphorylation. Now this is going to happen along these folds called cristae. This is the inner mitochondrial membrane. I'm just going to label that. Inner mitochondrial membrane. Okay. This is the outer membrane, right? Outer membrane. And the space in between those two, this space in between is called the because that's between membrane space, okay? So this terminology is going to be important that you can kind of keep up with. All right, now these folds are called cristae, so I'm just going to call this a cristae, right? That's plural. So all of these cristae, the folding makes it so there's lots of surface area. That's very important. All right, so along the cristae is a series of proteins and enzymes and other things that are going to be responsible for oxidative phosphorylation. So I'm going to show you just in one place, right, where we have something called an electron transport chain. Okay, so this right here is called electron transport chain. Okay. So this is a series of proteins along the inner membrane, and these are going to be passing electrons down this electron transport chain. And where it gets the electrons, so we're going to have all these electrons pass down, and the electrons come from all of those electron carriers, right? So all the NADH plus H pluses are going to drop off an electron here in the electron transport chain. So the NADH plus H pluses will actually, at this point, they will be oxidized. And they're going to give an electron here. I'll do my electron in purple. How's that sound? They're going to give an electron here. And this electron is going to be passed down the electron transport chain from one molecule to another. Now, each of the molecules in this chain are more electronegative than the one before it. So that electron freely passes down, just like it's moving kind of, along its gradient from one to the next to the next. And that energy of the electron moving down this electron transport chain, that energy is going to provide the power to pump hydrogen ions into the inner membrane space. So see all these hydrogen ions? So the electrons coming down the chain, passing from one molecule to the next, each molecule in the process is more electronegative than the other. The final acceptor actually is O2, oxygen. This is where the oxygen is necessary because we know that oxygen is really electronegative. So this is where our oxygen, this is going to accept the final electron is an oxygen. It's actually half an oxygen, right? And we're gonna actually take that oxygen and add a hydrogen to it, and we end up getting water from that. So because there's hydrogens in here too. So this is where the water comes from. You know, if you remember the equation for cell respiration, I'll just do the equation right here. We've got C6H12O6 plus O2. So here is where the O2 goes, right? And then the product is going to be, of course, we've got ATP, but we're going to get CO2 plus H2O, okay? Here's the H2O. And the CO2 we know came out in the Krebs cycle. Okay, so now, electron transport chain, all the electrons are going down, going to be accepted by oxygen. And then the energy used to move those, you know, as it's moving down, will pump a whole bunch of hydrogens into this inner membrane space. Okay, so this intermembrane space is important. Now, that means we're going to have a super high concentration of these hydrogen ions. And they are going to want to diffuse along their concentration gradient, but the only place they're going to be allowed to do that is through an enzyme that's embedded in here called ATP synthase. Okay? And the hydrogens will only be allowed to migrate back to the matrix through ATP synthase. And as they're moving along their concentration gradient, so these protons moving along their concentration gradient, that provides energy to do work. That's a very important term. That's called chemiosmosis. Okay? So the hydrogen ions are going to move along their concentration gradient to do work. That work, in this case, is going to be to build ATP. The protons moving along their concentration gradient to do work is chemiosmosis. As they move through ATP synthase, what ends up happening is that ATP synthase takes ADP plus a P, and sure enough, it's going to make ATP. And it makes a whole lot of ATP. In fact, for every NADH plus H plus, you get 3 ATPs. So for NADH plus H plus, you're going to get 3 ATPs. So I should, right? And the FADH2 is interesting because actually the FADH2 doesn't get passed to the top of the chain. It actually comes partway down. So for every FADH2, you end up getting 2 ATPs. Okay? So in the end, you can take all of your NADHs and multiply that by 3, and your FADH2s, multiply that by 2, and that's how many of the ATPs you're going to get in the end. And essentially, it turns out to be 32 or 34. We say 32 or 34 because sometimes the link reaction, it might pick up an FADH instead of the NADH, so it can vary a little bit. But that's why we get so much ATP out of this process, okay? And the whole thing is called oxidative phosphorylation because oxygen is driving that electron transport chain, the movement of those electrons. So we have to have oxygen to make it happen, and the phosphorylation is, of course, making is phosphorylating that ATP. So we're making ATP with the use of oxygen, right? And chemiosmosis is also some power that's necessary to get that through. But that's because the oxygen is powering, creating this concentration gradients between the hydrogen ions that are going to flow through here and help drive the ATP production.